Adelante.
Hi everyone. My name is Daniel, and I'm working in Paco's Barona's lab. I'm gonna share my presentation now. I'm currently at my 4th semester of the program, and I'm gonna talk about the role of microbial communities associated to symbiotically facultative Nostocales. But first, I'm gonna briefly mention more well-known nitrogen-fixing microorganisms, which are Rhizobium, Bradyrhizobium, and Frankia, which form symbiotic relationships with some plants, but in a limited manner. On the other hand, we have multicellular Nostocales cyanobacteria, which can form a facultative nitrogen-fixing symbiotic relationships, for which two types of cells are usually necessary. One is the heterocyst or nitrogen-fixing cell, and the hormogonia, the motile form of the filament. This bacterium can form nitrogen-fixing symbiotic relationships with the aquatic Fernazola, with fungi to form cyanolichens, with the angiosperm family of Cunera, with many bryophytes, but most importantly with liverworts, homeworts, and mosses, and finally with cycads. In these systems, the host usually attracts the symbiont from its surrounding and host them in specialized organs like the coralloid roots in the case of the cycads. And even though the molecular mechanisms involved in the establishment of this symbiosis changes from system to system, it has been shown that the same symbiont can infect multiple hosts. So, for example, you can take a symbiotic organism isolated from Gunera and use it to infect a bryophyte and vice versa. We call this ability symbiotic plasticity, and many symbiotic strains isolates from cycads, from biophytes, from Gunera, and from fungi have this ability. Furthermore, this symbiotic plasticity is found in this conserved clade of Nostocales species, which group together regardless of their origin. So here we have nostocs isolated from mosses, from other bryophytes, and from cycads, and which indicates that this is a phylogenetic trait. So we ask ourselves, what makes symbiotically plastic Nostocales different? And we think that it's their ability to form bacterial interactions. Why? Well, because we look at previous studies on this subject and found that there is a conserved cohort of bacterial taxa that includes the Nostocales associated to the symbiotic niches. We also found that the specific associations between taxa with Nostocales have physiological effects on these bacteria. For example, the associations between nostoc and Rhizobium affects nitrogen fixation, nostocs nitrogen fixations, and in the symbiotic system of Azolla, we have Rhizobiales. Well, the Rhizobiales were found that have conserved the nitrification activity. In cycads, the specific association of Nostocales and Caulobacterales involves the production of a natural product called indigwidin-like, and in cyanolichens, Sphingomonas and Nostocales were found to be co-localized. Finally, whole bacterial communities that contain a combination of these three groups can promote or repress cyanobacterial growth. So the hypothesis of this project is that Nostocales bacteria interactions enhance Nostocales' ability to colonize and interact with the host, which may contribute to symbiotic plasticity. So the objectives of the project are to construct a phylogenetic framework to place cyanobacterial isolates like the one I've shown in the introduction, find the functional and taxonomical bacteria core shared between cycads, Cuneras, and the bryophyte Plasia, find functions and species that could affect Nostoc's ability to form the symbiosis, namely its ability to move towards the host and to fix nitrogen, and test them both in vitro and in vivo and in vivo system, an implanta system. So to do this, I'm proposing this method. I'm using this methodology, which begins with the sampling of symbiotic communities from cycads, from Gunera, and from Plasia, which we then take back to the lab and which we co-culture in the lab. And then we can get isolated strains, and also we can use those co-cultures to make metagenomic analysis, which we then use to find functions that could be interesting. And then we take the isolates and we put them together to make screening, to make a test of interactions between the cyanobacteria and the bacteria, to see changes in Nostocales to test Nostocales to fix nitrogen and to form Hormogonia. And then we take those synthetic communities and we plan to use it in infection assays with the bryophyte plasia. The screening interaction studies are made with isolated cyanobacteria and an isolated bacteria, and we want to measure the changes in nitrogen fixation and changes in motility, because both of those things are very important for the establishment of the symbiosis, and also because we can see easily changes in both of these things because they are associated with a certain cell type, in the case of nitrogen fixation with heterocytes, and in case of the motility to the Hormogonia. We're also going to use just one isolated cyanobacteria to diminish the number of combinations possible between cyanobacteria and bacteria. And we are using plasia as our model organism because it's a well-established model organism used to test Nostocales to form symbiosis, and it's also very easy to view to see if the association is being carried out, like in this photo over here. So far, we've isolated symbiotic communities from 11 different cycads in 4 different locations, which are Querétaro, San Luis, Guanajuato, and Veracruz, and 5 Punera tinctoria plants from Valparaiso. We've made stable co-cultures from this sample plant, and we sequenced the 11 cycad co-cultures using shotgun metagenomics to find out more about the taxonomical and functional composition of these co-cultures, which, as we can see in this flowchart over here, involves the taxonomic classification of reads, the assembly of the metagenome, the posterior binding or grouping of the contigs, the reassembly of these of individual genomes, and the functional notation of this metagenome assembly genome, or MAX. So, beginning with the taxonomical profiling, we did the taxonomical assignment up to the level of reads using 2 programs which give us the identity and abundance of each order present in the sample, which is something like it can be viewed like this graph over here. And when we took all of them to Caulobacterales and Rhizobiales abundance is it's higher on samples from Querétaro, and we showed this. We found this by performing Krustal-Wallis test to check the significance of changes in abundance between the samples from Querétaro and elsewhere, and then we proceeded to the taxonomical assembly of the reads, for which we used metagenomic specific assemblers, and we choose the best assemblies based on the resolution of discrete coverage groups, which will help us to improve the quality of subsequent binding process. And this is basically what I was saying. As we can see in these graphs over here, there are discrete peaks in coverage, which indicates the presence of an individual isolate or an individual species, which we can then reconstruct. Which genome can we then reconstruct? And we choose the assemblies which have the better graphs. And after getting the beans, we proceed to the functional notation. And in the Nostocales beans, we saw the presence of the necessary genes to fix nitrogen, to develop heterocytes and form Hormogonia in most of the samples, except in this 4 over here. And in the rhizobiales beans, we didn't find any nitrogen fixation genes or nodulation genes, but we found the nitrification genes. Well, some of the nitrification genes. We also found that there is a lot of chelation and metal uptake genes, which is really interesting because this function is really necessary because the nitrogen fixation requires a lot of metals. And we found that xanthomonadales and rhizobiales beans are enriched in siderophore synthesis genes, which is an interesting thing to look into the co-cultures. And now, going back to the bacteria, we are using a battery of 8 different culture media to obtain isolates from the interaction experiments. And we are also working on isolating a cyanobacterial strain, which we will use to make the interaction experiments. So as I mentioned, the nitrification genes were found in the Azolla symbiotic community and in our cycad metagenomes. So we searched this enzyme from this pathway through PCR, and we found that there is a common that 9 cycad isolates have the NIR-K and NIR-S genes, but that only the 4 cycad isolates that came from the Querétaro sampling site have the NOS-C gene. We have also been working on an easier way to quantify individual production because before, which is through HPLC, because before we were looking at it through fluorescent microscopy. So this is a much better way to do this and in a more quantifiable way. And well, I was planning to do the correlation experiments, the association experiments, but I couldn't. So this is like a summary of what I've done so far, which include the parts that I'm currently doing or trying to do as well as I can, and also the parts that I need to finish before getting to the part of actually now combining our cyanobacteria with the isolates to test at least these functions. So far we have that the cycad population from Querétaro is taxonomically enriched in rhizobiales and caulobacterales, that symbiotic rhizobiales and xanthomonadales appear to be enriched in siderophore synthesis genes, and most of the cycad symbiotic rhizobiales have the nitrification activity, although NOR-C appears to be found exclusively in Querétaro samples. With that, I just want to say thank you to my lab who have helped me to do many of those experiments and who have also been helping me remotely now, and to you for hearing me. So thank you.